different tools. These are different tools basically, and you are all young budding minds, and we would like to introduce to you the way the modern tools have revolutionized agriculture in all facets of life, right? So suppose I say today transgenic plants, it evolves a different response from environmentalists. But do you know that all the insulin that most of your elders who are diabetic are consuming is transgenic? It was 10 years hence, earlier it was to be extracted from the pancreas of horses, right? But everything today is transgenic, and we don't have an iota of what you call as repercussion against them, right? And nothing has happened to any one of you or any one of your elders or any one of your known things. Science is for betterment, right? And these are also the tools. I'm thankful to my co-authors who have helped me develop this module, Supriya, Sneha and Patil, or Jain from NRCPB, and Manish and Rajiv. Now when we are looking at improvement strategies, the whole idea is to develop a good germplasm which has the qualities that we would like to have. It can be that we are looking at genetic resources, or we are looking at genomics to create a variability. Variability for what? For the genes under consideration. And when we were going into the earlier methods, we were looking at conventional selection. It's a beautiful saga of a journey that has moved past. And if you look, when you look at selection by Syrians, they were doing selection of date palm just by visual, right? Then over slowly years you had the mass selection, okay, where in a bulk of people you were selecting individuals. Then again over years biometrics have come, or use of mathematical techniques for selecting plant types. And at the same time there was a fly group, the Morgan laboratory, which was trying to decipher the genes, right? So this was the era of quantitative genetics, 1940s, Sir Ronald A. Fisher. As we moved, we had Maheshwari and Guha who started the pollen culture, led to pollen sorting, and in 1990s, the transgenic era. At the same time, at the other end, we had the molecular marker era, right? Basically, all these are tools. Tools for the person who is going to use either in physiology, either going to use in pathology, or going to use, it is across the animal systems or the human systems or the plant systems. These are various tools that help you to discern the character of interest best. And why do you want to improve this character? So that you get an improved germplasm. This is the basic thing. So if you look at the plant breeding components, basically we are into a plant breeding course. So if you look at the plant breeding components, what we are trying to look is whether it is the molecular breeding tools, or whether it is the quantitative tools, or the Mendelian tools, or the cytogenetic tools. The whole idea is to aid you in developing lines or varieties of interest which have greater economical use or aesthetic use, isn't it? This is the goal of plant breeding, or this is the goal of crop improvement. So what you are trying to do is better predict the phenotypes from the genotypes, right? You want to actually estimate what your phenotype would like to if you are using these two particular genotypes in the crossing program. Earlier we based all our readings on visual selections, then we used metrics for the quantitative traits. Now we are trying to use molecular markers. My particular lecture would lead or look into the molecular marker use in the crop improvement, particularly of chickpea, isn't it? So what can this Ciceromics, because it is chickpea genomics that I am speaking of, would help you? Well, it would help you to have a comparative study. All this notes is given to you, you needn't copy it, and it will be given as the slides also would be presented. Just enjoy the story, okay? It will help you to understand what are the comparative maps, and it's very important. Earlier, all the crop improvement people would used to look at Arabidopsis and Drosophila, isn't it? But now we have within our melilotus, means lotus, you have it, isn't it? So that is more nearer. This is how comparative maps allow you to look at genes or gene complexes from your own systems or in the systems that you are going to work with. No means a human and housefly have 99% collinearity, right? So the second one is you will have markers which are also of course rapidly evolving and which would tell you how the narrow domesticated germplasm evolved from the wild species. The third is you would able to develop the maps. You can use this information for studying various patterns. Now if you are looking at genomic assisted breeding in chickpea, or in short you are looking at applying the genomic assisted breeding in the crop of your interest or in animals or in humans, what you require? You require a robust breeding program. You require a sound breeding program. Finally, you want to develop a variety, you want to develop a pedigree horse, you want to develop a pedigree dog, or you want to develop a superior chickpea line. So you need to have a robust breeding program that actually breeders can do it. Then nothing can supplant a large scale field screening. Something if you think this is a wizard that would work and you just put the marker and you get the line, certainly not. You need to identify it and look at it visually. The third is to discern such molecular markers which are linked to QTLs of interest. You require well developed mapping populations. You have all these three, but suppose I am not habituated to use these tools. For me, if they are Greek and Latin, then they are of no use. So you require breeders, and it is better to catch them young, like we are trying to diffuse into you the modern technology so that you are caught at a young stage and it doesn't become something which you want to avoid, isn't it? Basically, you should understand these are basic tools. Of course, to develop such tools, you require a strong biotechnology and bioinformatics team, and most important that the people among these teams communicate with each other, isn't it? But you also require some money, so you require good projects, right? So what happens for us, for example, we have a very strong breeding network at IRI for chickpea. Among the country, we are the most leading chickpea breeding group who have released almost 33 of the 120 varieties. Almost 1/4 of the varieties were released from this particular center. We also have a strong molecular breeding and biotechnology team, and we try to interact between these two teams. Now the markers have been evolving, right? Like when I was a student sitting across in 1995s in these benches, that time isozymes were there and rapids started their journey. And then as we moved on, we have come to SNPs and DATs. With every changing marker system, the science is not changing. What you should know is you should know what you want to do with your work. Hannah Keats has very factly said that if you don't know which harbor you want to go, no sail or wind is useful, right? So you need to understand. For example, a rough thing is, we are a group of about 30 people. We have boys and we have girls. So let's say instead of RFFPs, I put the intervention as gender. How many groups will form? 2 groups, right? With the same 38 people who are sitting here. Now I change, let's come to RAPDs, I say state as the intervention. It's an intervention, you see, or a statistical treatment. Maybe we are from 6 or 7 states, right? The same 40 people, right? Now let us move ahead and we put age and as an intervention. Maybe 4 or 5 groups. Maybe job as an intervention, right? I am into a permanent job, we have research fellows, we have students, and we have technicians. Again the same 40 groups. And suppose I have used age as an intervention, and what's your good name? Neelu. Neetu has used age as an intervention, Sneha has used job as an intervention. The same set has formed different groups. Is the grouping technique wrong? No. Right? The same subjects group, but you should know for what you are doing. As a researcher, you should know for what you are using these techniques. For example, your group does not have, your crop that you are working does not have any markers, right? Any marker system developed, then rapids are the best bet, right? If you are looking at lineology, if you would like to look how the interrelationships in development has occurred, then RFLPs and AFLPs are best, right? If you are looking at where the genetic changes over evolution has worked on, the SNPs are best. So you should know for what you are working. No means no wind is useful, right? With the small introduction, we would be going into my work which we have taken up. See, basically we have been constantly hearing when I joined into the Chickpea program that the Chickpea program is very narrow in its genetic base. Most of the pulses were very narrow in its genetic base. Initially, we had a particular program with ICAR-IARI where we collected the land races. And then we had an access to the ICRUSAT gene bank where we could actually get all the lines. Now we started looking how diverse is this particular group. And when we started looking, we saw a beautiful pattern that has been shown by the Darwin's tree, where all those which were from the Indian subcontinent branched different from those that were actually in the what we call as Vana region, West Asia and North Africa, which is the golden crescent from where the chickpea has actually evolved, and branched differently along with the wild species. This actually led us to a particular program where we started pre-breeding. So we could understand that if we want to increase the diversity that is available, we need to cross the Indian subcontinent lines with the Vana region lines. So this would have happened because over years the breeders have sourced their germplasm from the major institutes of IAPR, IARI or JNKVB or from ICRISAT and started using them. And then let's say Neetu has developed good germplasm, I borrowed from her. So it has become something like a inbreeding program. Therefore, we have narrowed down the available diversity. That's what we have started, and of course we published this in Journal of Genetics, and then we could actually identify. See, if you are able to identify such wide distances, one is identification of wider germplasm, then utilizing them to develop a mapping population. This is what we tried to do. Okay. So then what we started looking is, because these were orphan crops, most of the molecular work has been done in case of rice, it has been done in case of wheat, in case of soybean, in case of tomato. And certain times it's a paradigm shift of your views. Can you know where was the first QTL breeding done in case of any crop in the world? Okay, where was the first transgenic crop evolved? In which crop commercially? Tomato, flower saver, and you will be surprised to know that the first QTL application is also in tomato, where they derived the seed, what you call as the fruit size and TSS from Lycopersicon pimpinellifolium, which is a small what you call fruited variety. So you need to first understand how these maps occur, right? Now at the same time, I would like to caution you that you need to use as many markers as possible. Like I explained to my children also, see how much is the marker size, let's say SSR markers, if most of your labs are using, they're 20 mer size. The forward and reverse, 40. If you have used 100 markers, you have used 100 into 40, 40,000. Isn't it? That is 40 kb. And what is the genome size of chickpea, which is a small crop when compared to others? 7.71 Meyer megabastiers. It is like finding a needle in a haystack. You are saying a needle and you are comparing and you are saying, yes, I got everything. So it is a caution that you should be able to use as larger a repertoire as possible. The second thing when people go astray when using maps is, developing maps and identifying is something and their utility is second part. Whenever you would have seen the reports, you will see that many people have used wild into cultivated, and this throws a lot of variation, right? But most of your markers that have been identified are skewed towards the wild. So therefore, what we started is trying to develop a linkage map using cultivated into cultivated crops. We of course published this particular map in Indian Journal of Agricultural Sciences, and this is the only second map that has been published in the world. The first was Cobos et al. So what happened is these markers can directly be used, but the marker repertoire was less, and what we started going ahead was to saturate this particular map. And therefore, we have used a particular group of parents. We have characterized the extreme drills. We are looking at yield traits. Basically, there was a very less yield gain in case of pulses, more so ever in case of chickpea. This is because over years chickpea was grown in marginal lands. Rain-fed poor farmers were growing it. So incidentally, for example, एक बार आप उसे मार के grow करते हो, it is okay. But constantly, generation after generation after generation, it has been marginalized. And as a result, what happened? It had adaptive gene complexes for marginal environments. And this adaptation, we have a beautiful paper by Dr. Behel, who was Deputy Director General too. See, in this process of adaptation, chickpea lost, or most of the pulses, not only chickpea, lost their productive genes and adapted for the drought tolerant or harsh environment genes. And that's what we were looking, how to get back these genes. For example, in our vested interest in tomato to select for edibility, we have lost the greater TSS, right? Now, the reason why we lost greater TSS was different. Why? Because the companies which were looking at syrups or syrups, what you pronounce as, so they were looking at more juice content, whereas Indians use it for culinary purposes. So we require solidness. So you need to look at the local breeding values when you are trying to develop a particular line. So we started characterizing these rills, and we constructed the what you call dense map, which had almost 957 markers. Again, in molecular biology reports we published this, and we could now we have chromosomes because we have a consensus sequence, draft genome sequence. But when we developed this map way back in somewhere, I think it is in 2011 or 12, that time we were still looking at linkage groups, and we could identify solid QTLs both for seed weight, for plant height, for basic other quantitative traits. Now we didn't stop there. We thought we need to saturate it because we had a new genera of markers that have come into play, and these were the SNP markers. So we started looking how we could, and we have done a GBS for this, and almost 1 lakh markers we could get. We could refine them, and we could actually anchor about 4,000 of these markers, and we could identify a large amount of QTLs, right, for various traits in almost all the chromosome for various yield parameters, and which has come in Nature scientific reports. See, this is one part of the story, that you are developing markers. Somewhere down the line, you will be seeing how we are using these markers in the breeding programs, and this is how we started with. We wanted to actually improve the seed size of Kabuli. See, if you look at chickpea, you have two types of varieties. एक आप लोग जो काला चना कहते हैं या देसी, है ना? And the one that is Kabuli, or the bees colored ones. So you have two different types. 80% of the production in the world is for desis, which goes for your basin or flour preparation, right? But 20% is for your culinary. Basically, we want our chickpeas when we eat it. Desi खाते हैं तो थोड़ा कटक लगती है. It's hard. It has more polyphenols. It has a thick rind. So we don't want to use it in our cooking media. But we use therefore Kabuli, which have greater sugars, right, which are soft, beautiful to taste, they have better organoleptic properties. But we want bold seed size, what you call as the dollar chana. So we were looking at how to do this, and then that's how we have started with looking at PG0515, which is a bold seeded line, and Goxi, which is again a ICAR-IARI line, which is basically from Ethiopia, and we started looking at molecular markers which are linked to for this. If you remembered, I have showed you 100 seed weight link markers. So we started using, and now we have lines which are in our preliminary yield trials, which are extra bold. That means 66 to 70 grams, 50 grams and above. This is the target that we are basically doing. So if you look down around 2010, unlike other crops, 2010, the first marker assisted program, rice, was released from this own institute by Dr. A.K. Singh, right? So at that point, the legumes were lagging. We were called orphan crops. We were just making our marker repertory, and we were at the stage of what you call as marker verification. That means we have had a large amount of markers that have been developed in different DBT projects. We were also looking at some trait mapping, and then we were at the stage of marker verification. But this was the time, that is around 2006, 2010, this is what, when some good marker maps have been developed. Now this is one point again where your students need to, you have targeted, let's say, one particular activity, what we call as a gap research. You see, you have so many good labs around the world, and they have floated so many papers, like one you have heard just few minutes before, Dr. Manoj Prasad. You know, they have identified beautiful. So you needn't reinvent the wheel. You beautifully use all this information to validate these things to use yourself. And where there is gap, so that is stop. Where there is gap between two labs, let's say Nitu's lab, I'm sorry because I took your name, and Ashwini's lab, where there is a gap, I concentrate my research on that. I don't know, this will help you also increase your publication, because these are the places where no research has been done. And it becomes easy, you have a prior information on these things. So what we have done is a stop research. That means we have the resistant genes where different workers, of course, that this thing, for example, Mayer or Tulu, they have identified different markers, right? Now what we were trying to do is validate a set of these markers. I removed that slide. We could validate about 12 markers and 5 which are very robust, and what we do is routinely use it in our breeding programs. And we have a wilt-sick plot, means a plot which has a higher load of the wilt pathogen, but that is for race 4 and race 3 predominant, and I am sitting in New Delhi. न्यू दिल्ली में कोई चना नहीं होती है, कोई फसल नहीं होती है. पंजाब में होती है, उसके लिए मेरी wilt sick plot is sufficient. You grow chickpea in Punjab and Rajasthan, but that wilt sick plot screening is sufficient. But where is the major area of chickpea? It is in central India, it is in northern erstwhile Andhra Pradesh or Telangana and Maharashtra, where you have a different races that are prevalent. So what I do, I molecularly profile the resistant lines and try to identify which of the lines have the different genes. Sitting at New Delhi, this is the power I have using the marker assisted technology. Okay, I can look into my lines and tell, okay, these are the lines which are promising. Because when I enter my lines into the All India Coordinated Research Program, what is done is they are not evaluated at Delhi alone. They are evaluated all the 22 centers. So one of the conditions in case of chickpea is they have to be resistant to wilt in that region at all to be considered for their next year of trials apart from yield. So you would like to have this, and of course this is another paper where we were trying to introgress from the wild species into 256, not only yield QTLs but also QTLs for wilt and Ascochyta blight. This is a pre-breeding program where it is under actually evaluation. So this is again one of my students' work. Of course, we have taken the help of my better half, Dr. Tara, to generate this graphical genotyping or GGT technique, but my PhD student is currently working. So you have heard these words quite often, MARS and MABC, marker assisted recurrent selection and marker assisted back cross breeding. Both places you are using markers which are linked to the QTLs of our interest. So when do you use a marker for recurrent selection? When do you use for a MABC? This should be known to you. I think Dr. Gupta would have told, he is our guru also. See, it should substantially give at least more than 15% variation. A marker, if it gives more than 15% of the variation of that particular trait, I prefer more than 20%, then it qualifies for me to be a marker that is sufficient to be introgressed using the MABC technique, right? If not, I push it into marker assisted recurrent selection. What is marker assisted recurrent selection? You have different QTLs which are governing less than 15% variation in two lines. You cross them, you get a population, you identify which are carrying your MARS or the marker assisted lines for this particular QTL, you again introgress them, right? You have different QTLs. You would have seen when I presented a table, there were 2%, 3%, 8%, 14%. So we intercross among them. Of course, we have now a greater benefit apart from MARS, genomic assisted selection, which I should be covering in the later part of this lecture. So now if you look down into this particular chromosomal complex of LG2 loci, so we have this TR19 and I think TA110. The flanking markers which are obtained from WR315, so we have used them for our foreground selection, and the rest of the background is in the line PUSA 372, which I wanted to improve. And if you look down, so if you look down slowly over time, we have developed lines, and we also identified some sources. This is a time shot photo that I have taken from the same place. So you can identify that we have also identified new sources in our Wilczek plot, which one of my research fellows, Sauliha, is now trying to identify if there are any different alleles. World over, we are using WR315 as the universal donor. So we would like to see we have different robust alleles. We could hit upon some markers which are actually being worked, but we wanted to identify some lines which she is trying to decipher. And this is a land race from Latvia. And you can look down, you would have seen the time shot. Earlier all the lines were there. Now only this line is there in that single field. Highly tolerant. See, it is tolerant. It need not be an yield giving character, but we need to transfer this tolerance to our related traits. Now if you look at the mass derived lines, we have developed a huge amount of one. We have 61 lines, of which we are into advanced varietal trial in chickpea using the MABC product, and this year they are into the final year of testing, right? So that is the progress. And then what we are trying to do is we are trying to also identify if we have any SNP marker that is available. For example, we are trying to validate this particular marker. Now this is one point of caution for young students who are other than biotechnology field who are going to take up. Someone publishes a marker or someone publishes a thing, you don't directly use it into your research. What you need to do is validate in your own set. No means you will be disappointed at the later end. It has happened with us when we started using TA96. And see, this is what we have entered. This is a susceptible check. This is the recurrent parent into which we are trying to introgress. This is PUSA 372, and look at the MABC lines which are totally resistant, right? So the second, the stress that we are looking, this is abiotic stress. We are also working on Ascochyta blight, which is in the preliminary stages, or the botrytis gray mold, so I am not touching it. The other one is the abiotic stresses, and if you look down here, the drought is #1 yield reducer in case of chickpea, the terminal drought. One, it is grown on marginal lands, but then it is not given any irrigation in the practical condition. Can we ameliorate the effect of drought as well as other abiotic stresses in the improvement of chickpea? That has been a second target for us. And then we started working on this. Now tolerance, I don't say resistance in case of abiotic stresses, is governed by different factors. Many people have actually given different parameters. When Neil Turner started working and giving the plant type concept along with Donald, Neil Turner has said that it is the transpirational efficiency, right, that controls the tolerance to abiotic stress, particularly drought. It is okay in case of rain-fed crops, in case of irrigated crops, right, where you have an inbuilt soil moisture and that which is able to reduce the transpirational efficiency by various mechanisms like leaf rolling or because of waxiness is able to better control the drought. But it is not so in case of, why, what happens if they are controlling? So if they are controlling that, it's okay, they are taking less water. But in case of rain-fed crops, mostly the legumes, already the soil moisture is very, very low. So some other characters need to be looked into. So you require physiologist who clearly establishes which characters are there. Then you require biotechnologists who will be able to identify the donor and the gene in it. And then you require breeders who would like to transfer that gene, and then you require the agronomists who would evaluate which of this particular genotype under what conditions would be giving the effort. But only problem in all this is the conventional approaches are actually very time consuming. So we have tried to look out at the various what you call as physiological parameters, and then peculiarly stresses are different in different conditions. If some of you are from South India, you know how it happens. We call something called as Sankranti or the Pongal, which comes around January 14th to 20th. Till then it is okay, but then the temperatures don't abruptly rise, they rise slowly. Maybe 1 degree, 2 degrees, 3 degrees. Of course, by April, May it is hot, but it does not abruptly rise. Means it does not go away from 20 degrees to 40 degrees in the South Indian conditions. So at this time, when the flower chickpea crop is at the flowering stage, it is able to still respond to drought by increasing its root density and root length. Now we come to the North Indian conditions, right? Till February ending or into the March first week, you have a 13 degrees temperature, right, 13 to 14, and by March ending it shoots up. In April first week it goes away to 38. That means the plant has only 14 days or 16 days or at the most 2 fortnights to respond to this increased temperature. Root no doubt plays a role, but it cannot play a major role. You require some other parameters, and that we could establish using this max minimum plot, that it is basically the relative water content and the membrane stability index that plays a major role. But then that's a different question. As a breeder or as a crop improvement person, what I am interested in? I am interested in yield per se. मुझे ये पता नहीं है whether that is coming from root or whether that is coming from leaf or whether it is coming from stem. What I want, that plant should yield better under drought conditions. So the drought susceptibility index plays a major role, and of course there are relative differences in the groups per se. And then what we started looking is, can we actually evaluate for this drought per se? And then we had a paper published by Dharmender Singh in case of what you call as lentils, where they have used an hydroponic technique to mimic drought in seedling stages, and they correlated that it works almost with a 0.8 correlation with that of the adult plant stage. So we started using this particular methodology because it's a rapid methodology. See, initially you have a large germplasm and you want to weed out. A good breeder is one who weeds out, not who collects. So you would like to weed out and have few lines on which you can take what you call as plethora of concentrated observations. So we started using this particular hydroponics techniques, could identify 2 parents which have been used for developing a mapping population for drought. We could develop, and then we have used these 2 characters, membrane stability index, and we developed a linkage map, right, to go forward the story. We could identify the QTLs, and then what we did is we followed a 2-pronged approach. Already there are some resistant genes that have been reported in various crops. We wanted to see how many of these genes are working in chickpea, and if we could actually mine these genes and use them. You get a genic based markers which are much better. Second is identifying QTLs. So this is again one of my students' work, that is Neeraj, who has worked on this aspect. So where, and Supriya also, Supriya is here. Yeah. So where we started using the genic based markers which have given various responses. These has already been reported by different workers, and what we did is we started sequence alignment, and we could identify that in the resistant genotypes you have different markers. All the markers don't play the same role in all the genotypes, and we could identify, for example, sucrose phosphate synthase pathway gene in case of Medicago, which was linked with relative water content. So it maintains the leaf turgor under drought conditions, right? Or SPS Medi, again it's a Medicago gene which maintains the membrane stability index. So this association of the candidate genes we could actually identify and look out how they play a role, what is the functional role that they play in case of drought, this thing. But then we were looking at a group of genes, for example, the ERF group of genes. This is a large family apart from the DREB genes and the NAC genes, the ERF group of genes which play a major role not only in case of drought but in case of most stresses, in salt stress, in case of cold stress. So we were looking what were these genes particularly functioning and how they were responding, and this has proved that these also play a major role in case of chickpea, not only in drought, in case of salinity. So what happened is we have a large group of such gene based markers, and let me say based on the root ontology, ICRISAT has developed a population ICC 4958 into 1882, and earlier reports, if you see Dr. Varshney's, they have reported more than 2000 markers which were linked to drought before we zeroed in onto it. So what happens is, okay, I joined this program in 2010 as I said, so immediately I brought about 750 of these markers indented, and I started using in this particular population, and to my surprise most of them were not working. Now this is where, see, the lab is very good, the person is a very famous person, he is the top 10 cited authors in the world, and he is a very close friend of mine, we have worked together. So we both sat and discussed where this problem can be. So I am not putting that slides here, here it is where we hit upon the idea of tilling, targeting induced lesions in genome. See, the best way is just forget Nitu is my guinea pig today. Nahin. Nikita, what? Nitu? Huh? Nitu. Nitu is from some other planet, right? She has come over, the object has come over and she is very fast in running, right? We don't know how she is running. She has about 8 appendages with which she is running. So what we would do? We would cut one appendage. Appendage 1 is cut, but still Neetu is running, right? We cut her other hand, but still Neetu is running, right? We cut her leg, she is hopping. We cut both her legs, no, she is not running. So we come to the conclusion that the legs are the one which makes Neetu run. Move into deep into science, what we have done, we have deleted, right? We have deleted certain segments. So the best way to know the function of a gene is forming deletion lines, right? The whole idea is to form, this is what you do. You know this particular chromosome, you start developing deletion lines, but for a breeder it takes 40 years. We had Dr. Sarkar here, who has spent 30 years of his research developing the maize deletion lines for all the chromosomes. These days we neither have the time nor the funds, nor I will be still at a scientist level if I am doing. So what we try to do is, we try to create these deletions using a technique which is mutation. No, mutations are random events, but you know, that is why I am saying you know which source you are targeting at. If you are looking at root zone, right? So you look, you don't look, what you give is you are given a deletion, right? So now you have a large amount of lines which are different deletions, there may be in leaf, there may be. So now you phenotype at least 10,000 of these lines depending on the deletion frequency that it gives. So you will get some markers which are present but that root part is absent. That means these are housekeeping markers, right? You have that marker absent, but still that plant is drought tolerant. So again that marker is housekeeping marker. है ना? आपका नाम क्या है? Amresh. Whenever I am seeing for the last 1 hour is with spectacles. Amresh IQ is 100. Now I would like to transfer this IQ. Now what is the marker I am seeing every time? Spectacles. I take out the spectacles, and because Neetu also has, I put on her, but her IQ remains 60. That means those spectacles are housekeeping marker or identification marker, but not the marker for IQ. And what happens, the biotechnologists with no malice, initially they do this thing. You have heard the technique SSH. क्या है? Substitute. So basically what they do, they grow the tolerant variety or the resistant line under normal conditions, and under stress conditions look out what are the, from cDNA identify the markers which are expressed only under stress conditions in the resistant line. But the susceptible line also tries to fight that stress. They do the same thing, they get a marker under susceptible lines which are only under stress conditions. Now they remove these markers which are obtained under stress conditions only in the susceptible parent from that of the resistant parent. And what happens? They say that okay, we got some markers which are only in the resistant line under resistant condition, like with him, that and these markers, but then you need to validate them, and that's what the stilling helps. So from the 700 markers we could just get how many markers better? 5 if I am true. 1, 2, 3, 4, 5. Only 5 markers which are actually linked to our QTL for drought under terminal drought conditions that had come from ICC 4958. Imagine 40,000 DAT sequences of Rajiv, 2000 published, 750 putatively linked markers, but only 5 have worked. We have again worked, and then we became only the second crop after pearl millet to establish a tilling library in case of chickpea. So what we started is using these markers, particularly the NCPGR21 or NC21, which was linked to this particular root volume and what you call as root depth under terminal stress conditions. And then we started using these as foreground and the other markers as background, and then we could develop. You could see this is just a BC2F2 line. This is the cultivar PUSA 362, PUSA 372, and we have a large number of lines where we were trying to introgress. And this is the ICC 4958. You can look at the root volume that we have, and then in case of using this particular NCPGR marker, we could actually increase the root volume. And then what we did is we developed a large number of these lines, okay, and we started evaluating them. And then we could get some lines which were actually even getting higher than their what you call donor into which they were actually introgressed. And luckily we started using the SNP marker for identification of its wilt tolerance. They were tolerant, and this is the first report in the country where a marker assisted product that was just released on October 4th by CVRC, Central Variety Release Committee. See, the rest of the marker breedings that we have in rice were the SVRC released products, and I am really proud that we could actually establish in releasing a marker assisted breeding program, first anywhere in world in legumes and first CVRC released variety on October 3rd. And we had a large coverage, either it was in ICR platform or whether it was on the ICRISAT or in various magazines. So another stress that we were looking at is salinity stress. I will just move fast forward for it. We standardized the screening techniques. Again, we used the hydroponics technique method. Also, we were collaborating with our field experiment with CSSRI Karnal. So every time why I am saying is phenotyping cannot replace any good breeding strategy. A large population, good phenotyping, and the ability to discard, not select, the rogues Rogues means any unwanted types here. So it is better to have 5 good children than 100 children, right? So it is better to have 5 good plants with your traits than having 2000 plants having one trait each. So that is the quality of a good breeder. We could identify some genes. Actually, why I am rushing forward is I wanted to cover this particular topic, the breeding cycle. And this is where the genomic selection is. What happens is, till now in the government sector or the CGR sector, we are not looking at how to reduce the breeding cycle, increasing the breeding utility. But the private companies not only use marker assisted selection, but they use genomic selection as a routine way. Now what happens is, basically what you are trying to increase in your product, the greater gain for your trade, which we call it as, all of you are agricultural students? No. Yes. And which year you are? MSc. तो ये तो बढ़िया है. तो that's great. So you would have heard about breeding value, है ना? The genetic gain of the progeny when compared to its parental or the generation from which it was obtained is the breeding value. Now this is dependent on selection intensity, which is generally 2.06 to 0.6. What is the accuracy of your ability to predict this selection? Earlier it was visual. We have used what you call as biometrics, and we have moved to the molecular markers. What is the variance? Means additive, what we are looking is removing the G into E component using the various stability models, isn't it? And then what are the years per cycle that you are going to do? So this is genetic gain over time or the breeding value, and what we are trying to replace this is genomic breeding value. Excuse me. Now in case of see, most of these principles again evolved from animal or human and applied to plants. If you have looked at path, we will say C.R. Rao 1952. And earlier it was done, but then we look at the thing. So for example, again no malice to anyone, your grandfather of your father would have selected a girl who is your mother, right? Did he select anyone or did he make a choice in the arranged marriage? He made a selection, right? And what were the conditions that look? अच्छा इनकी बेटी है, दोनों अच्छे हैं, अरे इनके संस्कार अच्छे हैं. So he was trying to remove the E component of bad and was looking for E component of good, and of course which घराने की है क्या है, so he was looking at genomic. But these were which selections? Visual selections. Now let us move to animal. In animal, for example, Holstein Friesian, yeah, very good milch cow, right? It has a milking capacity, that means it has been measured, right? So therefore you cross it. In case of dog breeding or in case of horse, the pedigree is looked, hai, how many races it has won or how many derbies it has won. Yeah, the mother has won 8 derbies. The father's products have won about, it's always female horses that run. It's the father's products or the sires have won almost 10 or 12 derbies. So the pedigree is good, you cross. Now what are you trying to do in this pedigree? What are you trying to do? For example, we are all humans, right? And all of us have taken agricultural sciences. We are all educated, irrespective of different caste, creed, region. So we have some common genes among us that made us a tendency to go towards agricultural education, right? See, for example, some group are very good towards nursing. All of us have empathy. The difference between empathy and service is when empathy turns into action, it is service. So all of us don't, we have empathy, but we cannot convert it into service. Therefore, we are not into the nursing field, we are into the agriculture field. So we have a common genes which are working among us. So I would like to capture this common gene pools from Sneha, from Ashwini, from Apurva, Nitu and Harish. Now some of us are extraordinary. That guy has 100 IQ. This girl can run very fast. Let's say she is good at informatics. So when you are converting these into plants or animals, one cow has higher ability to produce milk, one cow has the ability to have resistance. So, these are outliers with that particular marker, whose marker identification has been done, or the outliers. I would like to capture these outliers. So, I would like to capture all this molecular marker information, and once captured, using this information I would like to select from the population lines which are having at least 80 or 90% of these markers, and then use them parents for crossing. What your parents did in visual selection, what your grandparents did in visual selection, what the animal or the animal breeding genomic selection now is most common, animal breeders have done through pedigree analysis, or what the breeder has done through quantitative genetics, we would like to capture those markers, and that is what is genomic selection, nothing else. So, you capture this gene, the genes or the SNPs which are common as well as outliers, but since capturing, simple capturing will not pave any result, you need to validate whether actually they are predicting or not predicting, right. If your prediction accuracy is good, at least 80%, it is a boon to the breeder. Why? For example, some of you would have worked heritability or working heritability, yield related traits, low to moderate means 40 or 50. Moderately heritable traits 40 to 60, highly heritable traits 75 and above. है? So, if my prediction accuracy is 80% and more, then that is a good line, right. I needn't wait for carrying out all these biometrical tests at the later initial generation. What I do, let us say I have some lines, I identify this marker group in which lines it is present, take them, but this is not for advancement of lines, this is for identifying parents for crossing. So, I use it now because marker, the base population, the breeding value is at this level, you have tried to capture all the good breeding value of the genes that you wanted, and you have crossed among this, you have increased a 1% or 0.1%. Why I am saying this is, all of you would have come across ICAR-IARI in your different breeding programs, or in your classes they will say All India Coordinated Research Improvement Program on wheat, barley, rice, chickpea, pigeon pea. I know how they release the variety, if it is giving minimum 5% increase in yield in the second year of testing, right, it is released. Most, let us look at one of the ICAR-IARI, which is one of the oldest, wheat, 1950s. 10 years, I am not putting every year, 10 years gap, 50 to 60, 60 to 70, 70 to 80, 80 to 90, 90 to 2000, 10, 20, 7 years, 7 into 5, 35% yield increase should have been there, but none of the chickpea or wheat varieties give hardly a 2 ton or 3 ton over the original. Initially there was a gap, and this increase did not come alone from breeding, you understand this. There was agronomy that was playing a role, there was a stabilizing breeding of entomology and pathology that was playing a role, there was soil interventions that have played a role. So, there was a productivity gain, but how much is the genetic gain in it, right, it is a small fraction. Now, unless you increase the base population level for increasing the genetic gain, no greater quantum leaps are not possible. So, that is what we are trying to identify using these particular parents, particular selection, this is genomic selection, but you need to validate for each crop, right. Okay, we are a group of people, suddenly we have Gopal Krishna who is coming, who has let us say 120 IQ. So, I need to capture his SNP profile. So, the genomic population on which you train your markers is dynamic like a idiotype. It keeps on changing. The old goes away, you have already the marker repertory and the new lines to be added. And mind you, this genomic population is what? Useful lines. So, this is not a diverse population, it is a breeding population, that you should understand. The difference between a core and a genomic population is, genomic population comprises the elite breeding lines that have been developed in a program over years, and 10% of which is being substituted to accommodate the new lines so that their profile is, once you get their profiles, okay, these are the different aspects. once you get these profiles, what you try to do is you validate these profiles, and that's what we have done. We have used an 320 genomic population that we developed through various analysis using the ICAR-IARI ICVT lines, the trials that they send over 15 years. So we have used 155 Desi lines and 145 Kabuli lines. We evaluated under different populations, and we selected DART markers for our program and looked how many of them are actually, which model. You have different models. For example, you have the forest model, you have the regression model, you have different models. Which model? Every model does not fit at every place. You need to identify which model fits best for your genomic population or for your crop, and then we could see that the best prediction accuracy using this genomic model, we could get it for traits like drought susceptibility index, seed yield, and 100 seed weight. Of course, we published this in Frontiers. And then what we started is, we didn't stop there. Last year we were in our second year. What we did is we looked at what were the prediction accuracies of them basically in the field, right? So initially when we started looking at these, we found that the Desi predictions never predicted the Kabuli perfectly. Okay? That means we learnt a lesson that you need to make predictions separately for Desi lines as well as Kabuli lines. It will happen. These are all examples from my crop. So when you are working on different crops, you need to understand. Certainly, when we look at the prediction value, all our IRI group, these were the IRI lines, fell towards one side. That means my genomic population was skewed because we have used only the ICAR-IARI and the IRI group of lines. So what we thought now is that we will be including all the AICRP lines in our genomic selection. Of course, we got genomic prediction from F5 lines, and what we did is we started selecting the performances. We wanted to know on performances. So 200 lines I selected purely based on the genomic selection or the modern breeding method. But as a breeder, I make my own selections too, right? So these selections I make it only phenotypically. If you look at F5, it is only on F7 where you do the F6 you do the PYTs. So I was selecting the progeny from F5 to F6. I have made my own 200 selection based on the visual traits. Once you get them, means we make 3000 single plant selections. Once we get into the lab, we look at the seed traits, and acha hai, plant type is good, so I make around 300, put a PYT. I also put a PYT for the genomic predicted selection. Means this genomic predicted selection from F3 to F5 was done only solely based on markers. I didn't do any other selection, right, in them. And then what I see low here, the genomic prediction accuracy and the phenotypic prediction accuracy. I need to see here. Okay. For example, the blue is the phenotypic prediction accuracy, the red is the genomic prediction accuracy. If you look down, they are following the same trend, and the genomic predicted accuracy was more closer to the regression line. It was more closer to regression line. This was a difference. We have both the lines here. For example, the polynomial predicted, which is the black one, and here also you see the polynomial predicted, the black one follows the genomic selection trend. That means the predictions that are based on my genomic selection using the forest model or the random forest model perfectly fit for me. The random forest model may not predict, you may have to check which model fits in wheat, but then now I have this, and not only that, when you are looking, if you look, see I have made both selections, as I said, as a breeder, the visual selections of the 200 lines and the marker based selection, right, which we have selected both from the Wilczek plot. And if you look above 9, this has done accuracy of 9, that is 5% of the lines. And what is the accuracy here? 17, right? So if I combine up these three which I have used molecular markers, they themselves are 98% accuracy. हमें 4 लाइन अच्छे मिल जाएंगे तो बढ़िया है, सोचते हैं प्लांट ब्रीडिंग नॉर्मल में, बट इफ आई एम गेटिंग इवन Out of 10 lines, if I am able to get 8 lines, that is something different. 100, I am getting 98 very good lines. And of course, we are trying to improve our prediction accuracy because we thought we will now use the Kabuli separate and the Desi separate. We have used different platforms for merger sequences. We have used the DART-seq, we have used GBS and all. Now we are thinking that we will use the Affymetrix chips, and certain times maybe the pedigree of the IRI lines that I used were not included in the genomic selection set. I have used it at all based on the ICVT trials 15 years. So the better is to have all the parents that most of the breeders use to capture. If you not, then you will be skewed. For example, in human populations, you will be skewed towards the African, you will be skewed towards the Asian, you will be skewed toward the Caucasian. So this is very, very important that you do this. And this is how gains are going to come. Like I said, next year we will be releasing what you call as Kabuli or Desi wilt tolerant lines through marker assisted breeding selection. See, this is not my work alone and I will be failing in my responsibility if I don't acknowledge particularly my students and the research fellows on whose work this whole presentation or the module has been made. All those since 2010 have joined, whether it is Afzal, Akil, and the new group of people like Sangeeta, Soliha, Apurva and Umashankar and Akash, they are there in the group. All my students, including the blue ones, they are currently present. Neeraj, Harish, Jorben. I'm sorry. We have a large amount of Indian and the overseas collaborators, and this work is a culmination of all this work. Thank you. Any questions? And all this work we have done in our lab. I didn't put anyone's work.
Any clarification?
I'm sorry.
Sir, as we are saying that pulses are hardy, like chickpeas is drought tolerant. So in one direction we are saying that it's tolerant, and in another direction we are breeding.
It's only relative to other crops. Certain times we are in this paradigm, again with no malice to all. The moment I say, सबसे बीमारू स्टेट कौन सा है, आप लोग क्या बोलोगे? बिहार, right? But सबसे ज़्यादा सिविल सर्वेंट्स कहाँ से आए पता है? बिहार से आए हैं. Similarly, this is only in comparison with other crops. But as such, if you are able to induce drought tolerance into chickpea, it will be able to produce more in these marginal lands, right?
Sir, whether chickpea markers are applicable in other syntenic
This
crops?
is, everyone has to train his own horse. You need to cross-validate these markers. So if these markers are from the closer species, they validate better, but it is not so. It's not a general rule. You need to, that is why you have to cross-validate your markers.
Thank you, sir.
We are here for you. He has taken so much of pains, or the government is paying, so that you can understand. Please ask freely. No means ऐसा लगेगा मुझे कि सिर के ऊपर से गया जय राम जी की तरह, हैं?
Sir, after genomic selection, you validate the
No. Genomic selection is for selecting parents for crossing. If you are thinking of selecting elite lines, it is wrong. It is for selecting parents for crossing so that the base population's BV, breeding value, is higher than the previous one. This is the whole thing. That's what we do in case of humans and in case of animals, and now we are doing it in case of plants.
Sir, in genetic gain, even you altering the all components after cycles of selection, what is the best genetic gain we can see?
वैल्यू कहाँ से कहाँ तक जा सकती है? 0 to
Once.
1 is Eldorado, 0 is Nether. So you operate somewhere between that. So we look at 0.8 to 0.9. 1 तो बहुत मुश्किल है ना? Most ideal. All our hypotheses are based on that, or anti-null hypothesis 0. है ना?
Even a level of 0.5 is
That's
quite,
great.
quite good. Anybody else?
Thank you.
If you don't have, I think all are hungry.
Yeah, literally.
Yeah, please.
Yeah, please, I'm free. थोड़ा जोर से बेटा.
In spite of those SSR markers we have validated, how many in just we have identified DART and SNPs which we have pulled?
It's a huge repertoire. There are good number of papers, 40,000 darts, 1 lakh and odd SNPs. RAPDs are no longer used. I told you should know which your repertoire is. See, the marker repertoire is evolving. So you should identify for what. Maybe for routine use in the lab, SSRs are best because they are reproducible. You can convert a good rapid marker into RAPD marker into क्या marker? SCAR. And you can make your selections based on it. So it depends on for what purpose you are using. I can't use a what you call RAPD or DART sequence which captures less marker set for using a selection. Like I said, if I am using SSR 20 into 2040 maximum, so into 10, 100 even if you are using, I am capturing only 4K of 7.71 mega million bytes of chickpea. So you need to know for which system you are working and use the type of
That's
marker.
fine, sir, but how many, still I didn't got my answer, how many DART and SNPs we have got?
40,000.
40,000 DART and,
40,000 data. But you have SNPs which are more than 1 lakh.
okay.
That's what I was saying. But we have looked at SNPs, I said more than 1 lakh we have captured in the genomic selection. We are going through the 35,000 SNP array.
But ideally, sorry,
No problem.
ideally you are looking at as less markers as possible
Yes.
for the assay system for final selection,
Yes, sir.
because you don't want to spend too much on that. You have some key markers identified, then you go with that rather
So
than
we need to validate them again for foreground.
foreground for genomic selection.
Foreground is, see, foreground selection and background selection is for MABC, it's not for genomic selection. Genomic selection, let's say in this group, I am trying to capture all the marker profile because ये सब agriculture के हैं, and then elite marker of IQ for Harish for running of this thing, for bio, means I am trying to also capture the elite markers. So in this, the QTL markers are to be kept. That means the donor sources are to be kept. I shall be giving the presentation except for the last part of the genomic selection because it is still under publication. Rest all I should be giving to Dr. Gopal Krishna.
So we forgot to introduce we there and started. And in the genetic division, we have one of the youngest principal scientists in the form of sir.
Thank you.
Besides this, he has got a whole lot of projects which he has listed. And very recently, he has come up with a product which was released through Martest and backward breeding. They have released a drought tolerant chip. And it passes, I think this is
First
the
in
first
world in
in
country.